Gene expression is the conversion of information encoded into the RNA that changes to a protein. Gene expression occurs in two different places in the cell, which are both organelles. In eukaryote cells, gene expression occurs in the nucleus and then occurs in the cytoplasm. Transcription occurs in the nucleus and transition happens in the nucleus. Since prokaryote cells don't have a nucleus, all gene expression occurs in the cytoplasm. The nucleus is like the command center of the cell. It holds all the DNA and RNA inside the cell. DNA is also known as deoxyribonucleic acid, a self-replicating material present in nearly all living organisms as the main constituent of chromosomes. It is the main carrier of the genetic information in the cell. Cytoplasm is a gooey-like substance that surrounds all the organelles in the cell. The location of the gene expression in the cytoplasm is in the containing region in the prokaryote cells. Transcription is the process by which genetic instructions in a specific gene are transcribed into the RNA molecule. Transcription occurs in 3 steps that usually occur in a promoter region. The enzymes called polymers begin the formation of DNA from RNA to a DNA template and binds to a nucleotide sequence of DNA where RNA polymers initiate transcription. The second step of transcription starts with RNA polymers adding RNA nucleotides into one of the DNA strands. RNA nucleotide sequence then begins with the AUG combination. 3 of these nucleotides together form a codon. Translation is the making of protein. This is the main way to make major types of protein. Other ways of making protein are imitated from DNA to mRNA. There are 5 steps to translation. Step 1 starts with 2 ribosmal subunits joined together, tRNA and mRNA. tRNA is a class of RNA that carries amino acids into ribosomes and bonds with mRNA from proteins produced there. To start, a tRNA amino acid must be carrying methionine. Step 2 begins when the polypeptide chain is put together. In step 3, the polypeptide chain grows as mRNA moves beside the ribosome. The polypeptide continues to grow until it reaches a stop codon. The 4th step of translation is the end of the amino acid growth. Once there is a certain amino acid combination made, the polypeptide bond starts to break apart. A polypeptide bond is the primary linkage of all protein structures.
Oh, you left your calculator. Yep.
Type 2 diabetes is the most common type of diabetes in the United States. Over 90% of all diabetics have type 2 diabetes. Type 2 diabetes is caused by obesity and a lazy lifestyle. Those with type 2 diabetes are born with the genetic ability to receive it, but it is caused by the environment which the person lives in. Type 2 diabetes seems to come with many symptoms. If you get type 2 diabetes, then you have 2 defects. One is beta cell dysfunction, and the second is your muscle tissue is resistant to insulin.
This might be.
Symptoms of type 2 diabetes include being very thirsty, weight loss, wounds that don't heal, and extreme unexplained fatigue. Treatments to control type 2 diabetes include healthy eating, medication, insulin therapy, and all of these help your diabetes if you treat them. Distinct mutations were found in the PPARY gene. In some cases, severe insulin resistance, hypertension, and type 2 diabetes were found. Gene mutation in insulin. As you can see, there are three mutations that are highlighted. This is caused by cytidine mutation in insulin.